welcome to the panelists and thanks for being here. This panel discussion is designed to educate the people listening about the disease, the patient perspectives, and treatment options. So in addition, though, to Dr. Lin Lu and Dr. Conejo, we also have other panelists joining us. Doctors Alison Crawford of Regeneron is here, as well as Oliver Dorigo of Stanford University School of Medicine, and Dr. Hong Wang of Talic Therapeutics. We're going to start with a few questions that have to do with the patient perspective, then transition to questions about treatment options. First question, panelists. Why does diagnosis occur late in the process after the disease commonly has progressed to stage 3 or 4? Anyone can jump in.
I mean, I think there's a couple of issues, right? One is the lack of symptoms or the non-obvious symptoms that patients, I guess, don't have. You know, there's very subtle symptoms of some kind of gut irritability and things, and there's some pain, but it may be felt in a different area. So there's not very strong symptoms to let people know that there's something wrong. And then really, we've got a really dearth of really good antibodies to detect ovarian cancer. I mean, I think Yvonne had mentioned the CA125, which is the cleaved version of MUC16. It's one of the main markers now that we can really help us understand the progression of disease or response to therapy, but it's not a good diagnostic marker. You know, it's not clear enough. And even the studies, even a few years ago, combining with detecting HE4 as well as combining with ultrasound, we're really not able to detect the early stage 1 and stage 2. And I do think we need a lot more work in identifying biomarkers that are coming up earlier in these patients.
Yeah, Janice, well, first of all, thank you so much for having us in this fabulous symposium and discussion today. I agree with Alison. It's very difficult to discover these cancers early on. Simply, we don't have screening that reduces mortality, and we can only apply screening methods if we have a way of detecting early disease. Jonathan mentioned at the beginning that early disease is very treatable with excellent outcomes after 5 years. So we can cure most of the early disease stages. One of the issues is that this is a very sneaky cancer, and a lot of those cancers don't actually originate in the ovary, but rather in the fallopian tube. And that's an even more difficult compartment to image by ultrasound or any technologies. I think the most recent publication that summarizes data out of the United Kingdom has brought this home very well, that even in a very large trial, including hundreds of thousands of patients, in the general population, we simply don't, with CN25 ultrasounds or regular pelvic exam, have any screening methods.
Thank you very much. So Dr. Lin Lu did describe a number of the drugs that are available for the treatment options. But of course, I think one main one to start with is still the old standby of surgery. So could you speak to how those might be put together? You know, is surgery done first and then chemo, or opposite ways around? So could you speak to the real life and the practical options that patients would be given if they presented to you today?
Yeah, I can maybe speak to that. You know, I think that what has sort of been consistently shown over the years is that the completeness of primary surgical resection to a state of no gross residual disease, some people will call it R0, others will say just complete surgical resection, is a very strong predictor of patient survival. And so because of that, the use of surgery is sort of very foundational in the treatment for ovarian cancer. Now, it's not just any surgery, though. It's surgery to the point where there's no tumor visible to the naked eye. And so one of the big decision forks that we're encountered with when we see a new patient with possible ovarian cancer is whether or not we can surgically achieve that state of no gross residual disease at the end of surgery. If we believe that we can achieve that state, and there's a number of scales and markers and sort of guidelines that we use that will predict likelihood, but they're not perfect. Then if we think we can achieve that sort of no gross residual disease state, then we will usually bring somebody to the operating room and attempt that. If we don't believe that we can, then there's actually been a number of randomized phase 3 trials, mostly outside of the US, which have basically shown that giving an abbreviated course of chemotherapy, so a neoadjuvant approach of treatment with platinum-based chemotherapy and a taxane, can sort of chemically cytoreduce somebody, and then you can bring them to the operating room and then implement your surgical techniques and strategies to, again, achieve a state of no gross residual disease. And so the decision point, again, is when we see the new patient is whether or not we can achieve that no gross residual disease state after surgery. And then it's important to remember that even after the neoadjuvant therapy, where you chemically cytoreduce somebody and then you operate on them, there's still a period of chemotherapy afterwards, so the adjuvant setting, to sort of address the microscopic disease that remains.
Everyone agrees? It was very good.
Yeah, I do agree. Yvonne and I did fellowship in G1 oncology at around the same time, right, Yvonne? And yes. When we did fellowship, Janice, most patients, right, Yvonne, underwent primary surgery or upfront surgery, as we call it. Mm-hmm. And these were tremendously invasive, long procedure with many complications, blood loss, patients spent days in the ICU. The mortality was actually not so insignificant. We have now moved really into more neoadjuvant chemotherapy treatment. In the United States, about 50% of patients with ovarian cancer currently are treated with neoadjuvant chemotherapy in advanced stages. And we like to do 3 cycles in general, then take the patient, if they respond, for what we call an interval surgery. At that time, as Yvonne says, we need to make sure we can take all the cancer out. Otherwise, these surgeries don't have much benefit. In some institutions, including here at Stanford, we actually do what we call heated interperitoneal chemotherapy, HIPAC, at the time of interval debulking surgery. And that is followed then by 3 additional cycles of chemotherapy intravenously. In Europe, right, Jose knows this very well as well, the percentage of patients undergoing neoadjuvant chemotherapy is up to 70–80%. So the majority of advanced cancer cases in Europe are treated initially with a systemic approach and then surgery. And I think that predicting in the future, we will have more therapies that are gonna affect the tumor in general upfront, so that we're probably gonna move away from the very extensive surgery even more so than we have already done with interval debulking.
Thank you. I did want to bring that up because I mentioned in my introduction that I had a misperception about the cancer and what really is stage 3 or 4 cancer. And I suspect that many others also have this same misperception that the cancer itself is very localized. And so the surgery you're talking about, I believe, from the little, I mean, you do it, and I've only read a little bit about it, but if you could just comment on that it can, as you mentioned, be very invasive. It can be in all 4 quadrants. It can take 6 to 8 hours for a particular surgery. So it is not a trivial thing at all.
And I think that the best analogy, I mean, I don't know if it's the best analogy. One of my favorite analogies for advanced stage ovarian cancer is actually think of a bowl of rice and all of the little sticky grains of rice that stick on the edge of the rice after you finish eating the bowl, after you finish eating the bulk of it. That's kind of the situation after surgery. And then so imagine operating and trying to pluck off all of those tiny little grains and even all the little stickiness that surrounds it. It's really dependent on like what you can see with your naked eye. and whether or not you can achieve all of it. And just really, so that's why the surgeries are very extensive, and there's, like, like Oliver said, there's a significant amount of morbidity attached to those surgeries, and hence sort of a shift over time to a more neoadjuvant approach, with the idea of, again, chemically trying to shrink that so that your knife and your surgical techniques can better remove the things that are visible. So that's sort of where how ovarian cancer presents, largely in that kind of realm. And it's very, very sticky.
Thanks very much. I had that impression that it was quite difficult, and so we've covered the current treatment options, and I believe you touched on the fact that in order to decide on a best treatment option or plan for specific patients, really dependent on particular characteristics of the specific patient. So let's move on to the question about what new might be coming up. What do you see, given your experience and backgrounds? What do you think there might be available the next 2 to 3 years, so short term as well as long term, 5 to 10 years perhaps, in terms of new options for ovarian cancer patients?
So maybe I can sort of kick off some initial thoughts. I think that one of the things that we've really noticed just in the last maybe in the last 3, 4, probably 4 or 5 years now, actually, is the significant interest in genomic testing of the tumors, and really taking a much more personalized approach to ovarian cancer treatment. Number one, and then number two, I think that there's a greater appreciation for and there's a greater interest for identifying sort of hereditary cancer syndromes. Because, like you mentioned earlier, I know what Jonathan mentioned earlier, and what Oliver sort of reinforced, is that early detection portends better survival. And what we've seen is that when we started doing more and more BRCA1/2 germline testing to look for familial or inherited cancer syndromes, we started doing prophylactic surgical removals of the fallopian tubes and the ovaries. And the prophylactic removal of the fallopian tubes and ovaries have detected early stage tumors. And actually, that's a lot of those surgeries has actually provided a lot of the scientific information that's really solidified this this sentiment and this belief that and with confidence that the typical high-grade serous ovarian cancer probably originates from the fimbriated end or the distal end of the fallopian tube. And a lot of that supporting knowledge actually has come from a lot of these theoretically prophylactic surgeries to prevent cancer, but actually have in fact detected either very, very early stage cancer or even sort of very atypical cells that molecularly mimic cancer. So I think that sort of what the future holds are, number one, much more greater interest in prevention strategies. And number two, sort of a greater interest in sort of these personalized and looking for personalized approaches. I don't know, Oliver, if you...
Sure, Yvonne. No, happy to comment as well, of course. I think you just heard from Jose a fantastic talk about how you can actually identify very specific proteins, right, in those cancer cells that then become a target for therapy. And this is the type of research we need to support, because currently chemotherapy, as you know, it kills unselectively cells in the body and affects too many of the normal cells. And we need to understand that every tumor is different in every patient. And it's even so that different tumors at different sites within one patient have a different molecular phenotype. They might express different protein. And I'm curious from Jose to hear about how robust the expression of the FS receptor or the olfactory receptor 2 is in ovarian cancer. I think those are very important characteristics of tumor. But in that context, I think I agree with Yvonne. You have to make these treatment personalized. And I think we have already seen some initial data from particularly vaccine studies used in patients after they complete the first-line treatment, meaning chemotherapy and surgery. These personalized vaccines have shown some initial efficacy in the sense that they do increase the time to when the cancer recurs as progression-free survival. And I think in general, this is where we have to head. I'm curious to hear Jose's ideas about this.
Yeah, thank you. No, I totally agree. I mean, I am a hardcore immunologist. So in my small world, every thought about the future is cellular therapies, right? And cellular therapies can include TCR transduced T cells. DUNSI is already conducting trials targeting NY-ESO1 expressing tumor cells, and CAR T cells, Penn, Fred Hutch, Memorial are already working on that. I think in the current version of the CAR T cells, in which I am a big believer, we are going to see suboptimal responses. In some patients, they may be very significant, but in general, they will be suboptimal because the CAR T cell is going to face something that does not face in hematological malignancies, which is massive immunosuppression at tumor beds and the metabolic restriction of the tumor microenvironment, right? So for instance, a former graduate student who was depicted in the first slide of the video that we have seen published a paper in Nature. We are co-authors, but actually this is his work and Lori Glimcher's, showing that if you get activated tumor-reactive T cells and expose them to ascites, tumor ascites, they get frozen, right? But that's something that we can learn to target because that's the result of ER stress that can be pharmacologically co-targeted, and genetic engineering is progressing really fast. CRISPR is already a reality. And that's the way I envision the future. There is a lot of push at the industry on cellular therapies. So I hope that something will work against solid tumors as it has worked against leukemias, right?
Maybe I can also jump in here. Can you hear me?
Yeah.
Yeah.
Okay, great. So I think, so I want to go back to the theme that Yvonne actually introduced in her talk. And I think when I was listening to Jose's talk and I was nodding my head as well, I think the immunotherapy still holds great potential for ovarian cancer. I think I'm looking at the horizon probably in the next 5 to 10 years and thinking about what's the next wave of immunotherapy approaches that could come up. So I work at Untitled Therapeutics. We're a biotech company in the Bay Area, and we're focusing on novel platforms and approaches in IO. And one of the central scientific theme for us is, we really need to engage both arms of the immune system, both the innate and adaptive. And I think, I worked in this area since probably 2011, and I've definitely see the great success of the first generation checkpoint inhibitors, but I also seen some of the frustrations of some of the later T-cell targeted approach. I think the field today is really the recognition that we need combination in the immunotherapy space, and we need rational combinations. So one of the things that I found really interesting is how we target innate immunity and target some of the myeloid cells, which we'll hear later from Oliver, but also how do we combine B cells and the T cells together. So, we're actually very excited. We have a program that we're advancing into the clinic next year, and we're hoping to explore in ovarian cancer as well. And then this approach is actually to engage B cells and to activate the B cells and then to help T cells to work better. And in animal models, we're actually seeing very potent response in models where PD-1, PD-L1s don't work, or the models where PD-L1 failed, and then we can come in with the therapy and see eradication of tumors. So we're very excited. And I think really to enable this type of approach, we also need translational approach to understand the immune landscape in ovarian cancer better. I think that was the earlier question that was already asked, is ovarian cancer cold? How do we know which one, right? Kind of get to that personal level. I think there's a lot of great science. Actually, I was just speaking with a friend who's at Genentech, and her group have published a couple of papers in 2020 and 2021, looking at over 300 ovarian samples from the ICON study, advanced stage, to really understand the immune landscape. And they characterized the immune infiltrated, excluded, desert phenotype, and then use single cell RNA to really understand molecular interactions that could be different in these different types. And then they, I think really interesting evidence suggesting maybe some of the stromal interaction, the fibrocytes, as well as some of the chemokine interactions could be better in different types of immune landscape in ovarian cancer. So I think the next, really 5 years, we're looking at a lot more understanding of how we measuring the understanding of the from the patient, from the biology, from the cancer, and the new therapeutic modalities, approaches that both Yvonne and Jose talked about, and then testing them in a clinic in a rational way. And also, I think, both Yvonne and Jose, you've seen a lot of clinical trials. How we do clinical trials, I think we also need to innovate, right? We always start the very late stage in our phase 1, but we know that the immune system have evolved, very much from the beginning of the disease to that stage. And we know that chemotherapy, as Oliver spoke to, is a very blunt instrument. And how do we move these promising therapies faster to the earlier lines of patient in order to help, more patients? I think that's a big challenge for the field as well. But, really encourage you to be here today and hear all the great, talks, and then, engage with you and engage with the community. I think the future, I actually very enthusiastic, about the ovarian cancer and having new therapies coming online in the next 5-10 years.
Thank you very much. You've given me a number of questions to ask, but I don't want to go off of this question without giving Dr. Crawford a chance to hop in and give your views on long-term, short-term, new options that might be available.
Yeah, I think we all agree that the biggest area we're going into is combinations. And that was clear from Yvonne's talk, but I do think with solid tumors, it's going to be very unlikely that one single therapy is really going to be the answer. And what's exciting is, we have so many options now. It's not just chemotherapy. We have the anti-angiogenics, we have the checkpoint blockades, we have the small molecules like PARP inhibitors. And then, they've not been approved. There's other, lots of therapies not approved yet for ovarian that could be really worthwhile, like epigenetic modifiers, etc. But I do think the challenge there is really helping us understand what are the combinations to choose. And also, actually, beyond what combinations, also how do you dose them? I think that's a bigger question as well. Especially with IO therapy, there's a lot of toxicities involved. And the CD3 bispecifics and CAR T cells especially have this cytokine response, the CRS syndrome. So bringing in combinations, especially at the beginning when you're first dosing, it may really result in very high adverse events. So playing around with the sequencing of dosing when you're doing these combinations is definitely where I think we should be going and focusing on. But I agree with Dr. Wan as well. It's an exciting area, and what's fun is some of these combinations that really show benefit, you wouldn't maybe necessarily think that's the best combination, but then you can test these combinations and then understand how they're interacting. For example, the anti-angiogenics, they do have some immune-related aspects. The VEGF itself can increase Tregs, increase MDSCs, as well as normalizing the vasculature. And there's been some mostly preclinical, but some clinical data as well, that the anti-angiogenics can result in increased T cells and a bit of increased inflammation in the tumor. So there's really a possibility to bring in really kind of almost opposing or very different therapies that can really help each other and interact well.
Excellent. Thanks very much. We have just a couple of more minutes, but I will go ahead and ask one question. Maybe long answers though. Nevertheless, I think this is a very key question to ask when we have the full panel available. And that's, what therapy gaps or specific patient populations need additional focus and why?
I guess I'll jump in with my short answer. Is that I think one of the things we need to do a lot better is really, you know, is brought up by Jose that there are different types of ovarian cancer. You know, it's only 70% are the high grade serous. And we're treating ovarian cancer a lot of the time, and we're getting differences in response with PD-1 even. You know, I agree that we only have around 11% overall response, but some of them are complete responders. So what are the differences with those complete responders? Is it as simple as they have HR deficiencies and therefore they may have more neoantigens? There's a few of the complete responders have been clear cell carcinoma patients. So is there certain subsets that respond better to different therapies? I think that's a key area we should delve into more.
Yeah. And I would add very quickly, in my opinion, clear cell ovarian cancer is very much an orphan disease. And TCGA data sets, I mean, completely ignore anything that wasn't high-grade zeros. We don't even have data sets of those diseases, you know. I don't know why. I mean, yeah.
Janice, I'll give you a very short answer. I think for me, the two groups of patients at greatest need are patients that are resistant to platinum or in general chemotherapy. And then second, the population of patients that has tumors without any deficiency in DNA repair mechanism, or those patients that are proficient, as we call it, in homologous recombination.
Yeah, I mean, absolutely. I think that, you know, what I sort of like to summarize those patients is that, you know, in the last decade, in the last several years, there's been sort of pivotal type therapies, really transformative therapies, the PARP inhibitors, the angiogenics, the use of platinum. And there are pockets of patients that have really derived great clinical benefit from those therapies. But just as there have been those pockets, there are those patients that Oliver just delineated, those platinum refractory patients, those people who do not have those genomic vulnerabilities. They really have sort of fallen by the wayside almost, because they don't derive that great transformative benefit, and everyone sort of everyone sort of latches onto how transformative those new therapies are. So I think that those patients who don't derive that transformative benefit, they rarely really represent the greatest unmet need. And then also, sorry, one more thing. And then some of these rare epithelial tumors that Alison and Jose sort of mentioned, the clear cells, the mucinous, the small cells, etc. Those are frequently tumors that do not have these molecular vulnerabilities that are currently served by today's transformative therapies. And so again, they represent another significant unmet need as well. And so there's actually, I would say that just for the group to know that there is actually increasing awareness and recognition of just that population of rare epithelial tumors. And, you know, and I'm going to be shameless here. You know, so we actually started a rare epithelial platform study dedicated for these, for women with those types of tumors, specifically to try to address that gap.